All right, so this is the third part of our discussion of the reproductive system. Today we're going to be talking about the HPG axis in terms of female regulation, and of course how the HPG axis can regulate two different simultaneous cycles. Usually you just think of a female cycle as being, 28 days and, ends with menstruation, but there's of course a lot more to it than that. And so this lesson is going to dive into all of the different hormonal shifts, what's happening with the developing gametes, the developing follicles, and the endometrium, the lining of the uterus. Once we understand the different hormonal and other types of developmental features that are occurring, we'll talk about birth control and why that works, and then we'll end today's talk with fertilization. All right, so this is a graph of the various concurrent cycles occurring within the female reproductive system. Generally, this cycle is referred to as a 28-day cycle, but in reality, only about 10 to 15% of women actually have a true, consistent 28-day cycle. For the most part, the majority of women have more variable cycles or longer or shorter, depending on who you are. All of these cycles that we're going to be talking about, we are going to talk about in terms of 28 days, but keep in mind that particularly half of this cycle, the first half, is the variable half. Okay, the beginning point that we are going to discuss, and of course it is a cycle, we can in theory start talking about the cycle wherever it is a circle, but we are going to start talking about this cycle on, essentially day one of menstruation. And so just to give you a little bit of a preview here, we're going to be looking at two different hormones, the levels of these hormones on the first day of menstruation. Here are three more hormones that are going to be very important. This here is showing us essentially the thickness and the thickness of the endometrium and essentially what is happening in that lining of the uterus. And so this portion right here, right? So day 1 to up to day 7 is menses. So that is menstruation, the shedding of the endometrial lining or the period. Okay, so the beginning of the cycle is day 1 of menstruation and not the end of the cycle as you might otherwise be familiar with. Okay, so that's just arbitrarily where we're going to start our talk. Okay. We can talk about these cycles in terms of development in a couple different things. First of all, we can talk about what's happening in the follicle. Okay, so remember that the follicles are held within the ovaries. They essentially sustain the development of the oocyte. Right, so from a cell that's arrested in prophase 1 all the way into metaphase 2. So the follicle is going to regulate that and essentially nurture that developing cell. And also it is going to turn into the corpus luteum and maybe even the corpus albicans, as we'll talk about today. So this series of events is one cycle, right? So we can talk about the cycles in terms of what's happening in the follicle. We can also talk about what's happening in the uterus, right? So this is occurring in the ovary. This is occurring in the uterus. Again, this is the endometrial lining. Okay, all of these cycles, which of course are happening at the same time, are regulated by the HPG axis, the hypothalamus, anterior pituitary gland, and gonad, so the ovary itself. And remember that the specific part of the ovary that is secreting these hormones like estrogens, inhibin, and progesterone is actually the follicle. It's not really the ovary itself, it's the follicular cells. Okay, and so we're going to see that there's a lot of talking back and forth between what's happening in the uterus and the ovary, and of course, communicating with the brain. Okay, so the next few slides, we are going to be walking through each of these different parts of the graph, right? So this might remind you of looking at that Vigor's diagram from the cardiovascular system, right? We looked at the ECGs and we looked at the pressures and the volumes and all those different things happening at the same time. Similar kind of concept here. All of these different features of the graph are just trying to show you how the different events are connected to each other. So we're going to start our conversation by looking at what's called the ovarian cycle. So this is really focusing on what's happening within the ovary. And of course, the ovaries hold the follicles, right? The follicles themselves are what kind of drives this process, but we talk about the entire ovary. Okay, so the ovarian cycle is the top half of the graph. Remember that we always start with essentially the first day of menstruation. So that's day 1 or day 0 here. In a 28-day cycle, the midpoint is day 14. And that's actually a really important midpoint. It divides the ovarian cycle into 2 consecutive phases. Each one is 14 days, at least in terms of a 28-day cycle. The first half, so the first 14 days of a cycle is called the follicular phase. On average, this is 14 days long, right? But of course, there is a lot of variability. And of the 2 different consecutive phases, this is the one that can be 14 days, it can be 10 days, it can be a month long, depending on the woman. Okay. It is during the follicular phase that we go all the way from this primordial follicle with one single layer of follicular cells to increasing the number of granulosa cells. Okay, so lots and lots of layers, they become cuboidal and they start secreting lots and lots of hormones. Also, we see this differentiation of the thecal cells on the surface of the follicle. Right? We'll see that they are going to be important in secretion of hormones as well. Okay. By the end of this 14-day cycle, we can see the development of this fluid-filled compartment called the antrum, and that is signifying that ovulation is just about to occur. And so ovulation occurs mid-cycle, right? So if we're talking about 28 days, it occurs on day 14. Note that this is associated with a spike in hormones, and so that's going to be really important to us here in just a moment. After ovulation, the follicle is going to turn into what is called the corpus luteum. Okay, so the corpus luteum is made up of the same cells, the same thecal cells and granulosa cells. They're going to be doing the same kinds of things that they used to do. But now they are going to be secreting more and more and more hormones as this structure continues to grow. And so the second half of the ovarian cycle is called the luteal phase because this is the phase that is controlled by the corpus luteum. Right, so here we can see the follicle in purple. It turns into the corpus luteum, which is yellow, beginning the luteal phase. No matter what, the luteal phase is always going to be 14 days long. Right? For better or worse, this is the consistent portion of every woman's cycle. Okay. Note that the corpus luteum is going to continue to mature and grow, therefore secreting more and more of those same hormones. If fertilization does not occur, it is going to essentially die. It's going to degrade and stop secreting hormones fairly suddenly, in fact. And at that point, we call it a corpus albicans. Okay. Note that the hormones responsible for driving the development of the follicle itself are the gonadotropin hormones, that is LH and FSH. Note that for the most part, over the course of the entire cycle, LH and FSH are pretty low, right? A little bit is secreted, but really not that much. However, this is not the case around mid-cycle. So around day 14, we see a spike in luteinizing hormone as well as follicle stimulating hormone. And this is going to be important because this spike in hormones is what triggers ovulation. Okay. And so let's talk a little bit more about how the follicle is functioning. Remember that the primary follicle, right, once it has been selected, is going to start making more and more granulosa cells. So those cuboidal cells who are really good at secreting lots and lots of stuff. Okay. In the secondary follicle, those granulosa cells, as well as the thecal cells that have differentiated in the follicle, are going to express receptors for LH and FSH. Just like we saw in the testis, certain cells bind to luteinizing hormone and other cells bind to follicle stimulating hormone. in the testis, Sertoli cells or cystinocytes bound to FSH, and the interstitial endocrine cells or Leydig cells bound to LH. We see the same exact thing happening here in the ovary, specifically in the follicle. The thecal cells, or the more superficial cells, are responsible for binding to LH. Okay, so when LH binds to the thecal cells on those receptors, what's going to happen is that LH is going to trigger a change within the thecal cell. Specifically, it is going to trigger the expression of particular enzymes to convert cholesterol into a hormone called androstenedione. Okay, and so this is not a term that you need to be able to produce for me. That's okay. But what I'm getting at here is that, let's see, a couple classes ago, we looked at that metabolic pathway from cholesterol all the way down to the glucocorticoids, to aldosterone, to testosterone, to estrogen. So all of these hormones are linked together, but it is the binding of LH that is essentially going to shift that entire pathway in the direction of androstenedione in the thecal cells. And so this is important because this is a precursor to estrogen. Okay, so remember we talked a little bit about aromatase. Aromatase is the enzyme that converts androgens, so essentially masculine hormones, into estrogen. Okay, so if we have the precursor molecule, androstenedione, and the granulosa cells bind to FSH. Okay, so here we see FSH binding to its receptors. It is going to trigger a gene expression within the granulosa cells. Specifically, aromatase is going to be upregulated. So we're going to make more of this enzyme that converts androgens like androstenedione into estrogen. And so in order to produce estrogen, the follicles need LH and FSH to bind to the thecal cells and granulosa cells, respectively. And so take-home message here is that we need both of these gonadotropin hormones to trigger the release of estradiol from specifically the granulosa cells. Now, also at the same time, inhibin is going to be released by the granulosa cells, right? So this is going to be part of that negative feedback loop to the brain to make sure that just the right level of hormones are produced at just the right times. Okay. Does that make sense? I know that's lots of endocrinology, right? All right. Okay, so remember that GnRH is released by the hypothalamus. That triggers the anterior pituitary to release LH and FSH. As we know now, LH and FSH ultimately trigger the production of estradiol as well as inhibin. Okay, remember that just like in the testis, levels or rising levels of these hormones are going to exert negative feedback on the brain, right? So the hypothalamus and anterior pituitary gland, so that FSH and LH levels remain fairly low. Okay, just because, well, we need them to be pretty low for most of the month, most of the 28 days, except for right before ovulation. And so here is that feedback loop here, right? GnRH is released by the hypothalamus. Anterior pituitary gland is going to release FSH and LH. LH triggers the thecal cells to release androgens, right? Androstenedione. And FSH triggers the granulosa cells to convert these androgens into estrogens. Right? Rising levels of estrogens as well as inhibin is going to essentially put the brakes on the hypothalamus and anterior pituitary gland, making sure that these two hormones in the middle remain pretty low. Okay? Note, if you look at the bottom of this diagram, it says early and mid follicular phases. Okay, so this is not always the case, right? We don't always need FSH and LH to be fairly low, fairly controlled. There is one point in the cycle where this entire process is reversed. Okay, so when estrogen levels are high, there's going to be a reversal of this entire process so that there is no longer negative feedback on the brain and anterior pituitary, but now positive feedback. And remember, positive feedback essentially enhances an existing response, and negative feedback maintains the responses or levels within a particular range. And so remember that LH and FSH, even at low levels, are going to be triggering the development of these follicles. Okay, as the follicles grow, there are going to be more and more thecal cells and more granulosa cells. Now, even if those cells are only themselves secreting a tiny bit of estrogen as we have more and more cells, the levels of estrogen are going to rise over the course of that 14-day period. And so we can see that even though LH and FSH are fairly constant, estrogens are going to increase over time. And again, this is just because the follicle is getting bigger, and each of these granulosa cells is going to be secreting the same amount of estrogen, but there are just more of those cells. And so usually rising estrogen levels is going to suppress the secretion of LH and FSH until a certain level of estrogen, a certain threshold value is surpassed. Okay, and so the actual level of this is not important, and this is surely variable from person to person, but ultimately the levels of estrogen are going to exceed some target threshold, and this is going to reverse the negative feedback and convert it into positive feedback. So now the follicle is, it is mature. Here's the mature antrum filled with fluid. The oocyte is now arrested in metaphase 2. It is ready to be ovulated. And so now that high level of estrogen is going to switch, and it is going to essentially tell the anterior pituitary gland to dump out as much LH and FSH as it has stored within it. And this LH surge, right, this release of stored luteinizing hormone from the anterior pituitary is going to trigger the rupturing of that existing follicle. And so here is the edge of the ovary. The follicle bursts and essentially spits out that secondary oocyte. Okay, here I have LH highlighted and underlined. The most important hormone for triggering ovulation is luteinizing hormone. To an extent, follicle stimulating hormone also increases. It's also important, but LH is the ovulation hormone. And so as we'll see farther along in this lesson, one of the ways that women can kind of monitor where they are in their cycle, if they're undergoing fertility treatments or trying the natural planning method of birth control, you can monitor luteinizing hormone, right? Whenever luteinizing hormone is very high, that is when ovulation is occurring. Okay, and so what exactly is ovulated? Well, the secondary oocyte, remember that this is a cell that is arrested in metaphase 2. Essentially, all those little chromatids are lined up. They are ready to be pulled into different cells, but not yet. And so I want to show you real photographs of ovulation occurring. So this up here is the whitest tissue here is the ovary. This is the follicle, and here we can see ovulation actually occurring. Right, so the follicle is going to burst and essentially spit out the egg, the secondary oocyte, which is surrounded by a couple different things. As we can see here, even within the follicle, the secondary oocyte is surrounded by a series of cells. We've talked about this a little bit before, but essentially these cells are granulosa cells that we are going to call the corona radiata. So this crown that radiates all the way around the egg, which is going to become important in fertilization. So stay tuned. And also there is what is called the Zona Pellucida. Remember that this was secreted pretty early on in the follicular development. It is a clear layer, and it essentially makes it really difficult for sperm to actually get to the egg. The only way that sperm can actually penetrate the egg and therefore fertilize it is to get through both the corona radiata as well as the Zona Pellucida. And so that takes very strong energetic sperm. It takes sperm that have the appropriate enzymes for doing so, have the right shape of the acrosome, et cetera, et cetera. Okay, so this ensures that only human sperm can fertilize human egg, and also that only one sperm can fertilize the egg. Of course, there are exceptions to this. Twins do happen, of course, but for the most part, humans have one offspring at a time. Right, so ovulation triggers the beginning of the luteal phase, that phase that is controlled and characterized by the activity of the corpus luteum, which of course used to be the follicle. Okay, the luteinizing hormone is not only going to trigger the bursting of the follicle, but also the conversion into the corpus luteum. Okay. The corpus luteum is going to function largely the same as the follicle itself, but due to luteinizing hormone and this further development here, the corpus luteum is going to secrete more of what is called progesterone. I've mentioned this before, but I want to say it again. Progesterone is pro-gestation. Gestation is pregnancy. So this is the hormone that is promoting anything pregnancy. Right? So all of the things that go into sustaining a developing embryo, getting mom's body ready to sustain a developing embryo, all of that is regulated by progesterone. And so we can see that progesterone starts to rise, right, over the course of this luteal phase because the corpus luteum continues to grow, it continues to be active, and so it secretes more and more of this hormone. Of course, if a fertilization event occurred, it would have occurred around this period right here. So the very beginning of the luteal phase. And so it is going to, the fertilized egg, right, the developing embryo, is going to essentially implant into the uterine wall, into the endometrium, during this phase right here. And so progesterone levels have to be really high to make sure that endometrial lining is maintained and it can support that implanting embryo. Okay, and we'll get into that in more detail here soon. The corpus luteum is going to live for 10 days. Right? Remember, this is the consistent portion of the cycle. It's always going to be 14 days. So 10 out of those 14 days, the corpus luteum is going to grow and grow and grow and start secrete more and more hormones over this time. Okay. Keep in mind that immediately after ovulation, negative feedback is going to resume. And so we can see the estrogen levels pretty much spike right before ovulation. Okay. And that triggers, of course, the unloading of luteinizing hormone into the blood. As soon as luteinizing hormone spikes, the feedback reverses again. So negative feedback is going to resume. Luteinizing hormone and follicle stimulating hormone are going to crash again, which is then going to decrease estrogen for a little while. Inhibin from the corpus luteum is going to, of course, enhance this effect, right? It's going to help this transition from negative to positive and positive back to negative. Okay. If fertilization did not occur around in this time, right, the corpus luteum is going to die after 10 days, right? So these cells are going to kind of shrivel up and they're going to stop secreting progesterone and estrogen and inhibin that they've been secreting all along. Okay, so in this graph, we can see that the corpus albicans is essentially the scar left over from this corpus luteum. And with this degeneration of the corpus luteum, we also get a concurrent drop in progesterone, right? The hormone that is maintaining the endometrium in order to sustain a new life. And of course, the other hormones, estrogen and inhibin as well. Okay, and so if fertilization does not occur, these hormones drop off after 10 days. Okay, so that by day 14, menstruation can begin. If fertilization and implantation occurs, the corpus luteum is actually going to be maintained. All right, so the end of this graph here would look a little bit different. This is showing us if there is no fertilization. If there is fertilization, the corpus luteum is going to continue secreting progesterone. Remember that we need this hormone in order to sustain the endometrium, to sustain the developing embryo. And so we really don't want a crash in this level of hormone if there is a pregnancy. Okay, so the corpus luteum is going to live for the next 3 months, so for the next or for the first trimester, essentially, until the placenta itself can take over these hormonal duties. Okay, it is the action of the follicle and then the corpus luteum that is going to determine the levels of estrogens and progesterone. It is estrogen and progesterone that controls what is happening in the uterus. So now we're going to transition into talking about the uterine cycle, or more commonly called the menstrual cycle. Again, this is directly controlled by estrogens and progesterone, which is controlled by the follicle, which is controlled by the HPG axis. Okay, so lots and lots of layers of control here. Okay. All right, so the endometrium, as I said a few slides ago, this portion of the graph is showing us both kind of a cross section of what the endometrium looks like. So we can see some blood vessels, we can see some glands, we can see that it is changing in thickness. Okay, so right after menstruation, it's really thin, but then it continues to get thicker and thicker and nice and kind of cozy for the developing embryo to implant. Okay. The uterine cycle is divided into 3 different phases as opposed to 2 in the ovarian cycle. The first few days is the menstrual phase. And so this is essentially the shedding of the lining, the destruction of a portion of the endometrium. All right, so this is when bleeding would occur. Remember that these graphs are just kind of averages. So it says days 1 through 5, but of course from woman to woman, this varies substantially from 2 days to 2 weeks, very variable. Remember that the first half of all of these cycles are the variable phase. This is the consistent one. Okay. In days 6 through 14, again, on average, this is the proliferative phase or the pre-ovulatory phase. And so this is when the thickness of the endometrium increases the most, right? So we have this proliferation of the tissue getting thicker and thicker and thicker, okay? Of course, leading up to ovulation at day 14. Okay, in days 15 to 28, always 14 days, no matter what. This is the secretory phase, right? So essentially, this is when the endometrium is secreting all sorts of stuff to sustain the embryo and to encourage implantation, to nurture that embryo, and to promote the development of this maternal fetal blood circulation. Okay, that is the placenta. Okay, in the proliferative phase, right, we're going to start here because this is essentially the growth of a new endometrial lining. This is stimulated and sustained by estrogens. Remember that estrogens are increasing because the follicle is getting larger and larger and larger, therefore secreting more and more estrogen. Okay. In response to estrogens, right, that is, the endometrium itself has estrogen receptors. When estrogen binds to the endometrial cells, it is going to stimulate mitosis. Okay, so it's going to start making more and more cells getting thicker and thicker. Also, these new cells are going to develop progesterone receptors. Right? Keep in mind that progesterone isn't going to be secreted until there is a corpus luteum. The corpus luteum is created after ovulation, and so progesterone levels rise in the next phase, in the secretory phase of the uterine cycle. But of course, the endometrium has to be ready to receive that message. And so it gets ready to receive that message by expressing progesterone receptors. That is, transcribing, translating, embedding within the membranes of these cells. Okay. Also during this time, the uterine glands embedded within the endometrium are going to start secreting essentially food. Okay, so we can see here that there are lots of glands made of epithelial tissue. They are going to secrete lots and lots of stuff, specifically a glycogen-rich mucus. Okay, so remember that glycogen is an energy storage molecule. Okay, it's really easy for an early embryo to just kind of break off glucose molecules, throw them into the mitochondria, and get lots of ATP as a result. Okay, so essentially, the entire proliferative phase is preparing for the possibility of a fertilized egg. Okay. Also, something else that I mentioned in the last class is that the cervical mucus is going to change. Okay, remember that this is the opening to the uterus. Here is the vaginal canal. Here is the cervix, the neck, the opening of the uterus. And so the cervix is lined with a lot of glands secreting mucus all the time. And this mucus is going to change over the course of the cycle so that sperm is essentially promoted, or sperm passage is essentially promoted, right? Allowing it into the uterus during this phase right here. Okay, so right leading up to ovulation and a little bit after ovulation, essentially promoting reproduction, and then the rest of the cycle, right? So menstruation all the way through way over here when the woman is no longer fertile, the cervical mucus changes to be thicker so that sperm entry is more or less prohibited. Ovulation happens, right, due to the processes we talked about in the ovarian cycle. This event begins the 14-day secretory phase of the menstrual cycle. Okay, so what happens in this phase is that progesterone and, to an extent, estrogen are going to stimulate further growth, further productivity of the endometrium. Here is the endometrium again. These uterine glands are going to get bigger and bigger and therefore increase their ability to secrete food for the embryo. Okay. Arteries are going to grow, right? So arteries grow and then recede every single month here. Here we can see that there is vasculature called the spiral arteries that grows up into the endometrium. Okay, so these are spiral arteries. Essentially, they are going to be delivering nutrients and oxygen, etc., to an embryo if it happens to implant within this wall. Okay, note that the endometrium is divided into the functional zone and the basilar zone. So the basal, the deeper layer, the basilar zone is going to stay no matter what, even after menstruation. The functional zone is what literally functions in supporting the embryo, and it is what is shed every month. Without embryo, that is. Okay. So these cells, of course, have progesterone and estrogen receptors, and both of these hormones, again, are released by the corpus luteum. Okay, and as the corpus luteum continues to grow over this phase, you get more and more progesterone and estrogen. Okay. All right. If fertilization does not occur within 10 days, right, and generally, the egg is not viable for that full 10 days. So if fertilization actually doesn't occur within 1 or 2 days after ovulation, this is going to occur. Okay. So without fertilization, no baby, the corpus luteum is going to degenerate, right? It starts to break down. And as a result, as I said before, the progesterone levels and the estrogen inhibin levels are going to crash. Okay. Without this continuous signal to support baby, support baby, support baby, now everything in the endometrium, or specifically within the functional zone of the endometrium, is going to start breaking down. Okay, so without the message to keep growing, keep building, the spiral arteries are going to kink and spasm. Okay, so essentially they are going to cut off further blood flow, right, leaving some blood kind of behind, trapped within them, and they're going to spasm. So essentially producing a little bit of movement to start dislodging some of these other cells. These cells are concurrently beginning to die, right? So all of these glands and everything are regressing back into the basilar zone, and all of this tissue that is more superficial is going to start breaking off, helped out, of course, by the spasming of the spiral artery, by contractions of the myometrium, right? So this very thick muscle layer down here, thus the cramping of a menstrual period or leading up to menstruation, that myometrium contracts, the spiral arteries spasm, and essentially all of this movement within the uterus is going to dislodge that lining. Okay. So this, of course, triggers menstruation itself, so the flow of this endometrial tissue and the blood within it. This, as I said before, marks the beginning of a new uterine cycle. Right? So you might think of it as the end of the previous cycle, but in terms of these graphs, in terms of this discussion, it is day one of the cycle. Right? Literally, the body, the ovaries, the follicles start developing on the first day of a menstrual period for the next menstrual period, right? No time is wasted. Okay. After the spiral arteries have constricted, they relax and open wide, producing this rush of blood. And so again, this functional layer is disrupted and ultimately it is shed. Okay. By approximately day 5, again, no time is wasted, the follicle, the new follicle for the next cycle, is already producing a lot of estrogen, which stimulates regrowth of the endometrium. So that's why we see the shedding of this layer here and then the immediate regrowth. Okay, so let's take a look at this big clunky graph again. Hopefully we can understand what's happening in it a little bit better. All right, remember that LH and FSH are both released from the anterior pituitary in response to GnRH from the hypothalamus. These hormones are going to bind to receptors within a developing follicle. That triggers the follicular cells to grow, divide, and differentiate into granulosa cells as well as thecal cells. Right? The follicle continues to grow, right? Ultimately producing an antrum here. This is a mature follicle with one single secondary oocyte arrested in metaphase 2. Now, these follicular cells aren't only kind of nourishing this oocyte, but they're also secreting estrogen and to an extent inhibin. Estrogen is going to exert negative feedback, which is going to keep LH and FSH pretty constant until a certain threshold level of estrogen is exceeded. And this is going to happen right before ovulation. Okay. All of a sudden, the negative feedback that estrogen is exerting on the anterior pituitary is going to be reversed, leading to a surge in luteinizing hormone. This LH is going to trigger the rupture of the follicle and the ejection or the ovulation of the secondary oocyte surrounded by the corona radiata and the zona pellucida. This oocyte is then going to be transmitted from the fimbriae through the uterine tube all the way into the uterus, and this takes about 5 to 7 days. Okay. After this brief surge in LH and to a lesser extent FSH, the negative feedback of estrogen is going to resume, and we can see a subsequent crash in LH and FSH, as well as estrogens. Now, these hormones, LH and FSH, also triggered the remaining follicular cells to convert into something called the corpus luteum. The corpus luteum is going to secrete both estrogen and, to a much greater extent, progesterone. Now, these hormones are going to trigger the further development of the endometrium. Okay, so estrogen rises here, triggering proliferation of the cells within the endometrium. Then we can see progesterone, to a much larger extent, is going to continue with this development for the next 10 days. If fertilization does not occur over those next 10 days, the corpus luteum is going to essentially degrade, die into the corpus albicans. Now, the corpus albicans is just the scar. It is not functional. It's not actually secreting progesterone. So we can see all of these hormones are going to crash at the end of this luteal phase in the ovarian cycle, as well as secretory phase of the uterine cycle. Okay, without this continuous stimulus to essentially maintain the endometrium, can go all the way back to the beginning of the cycle, and we can see that the endometrial lining, that functional zone, is going to be shed. Okay, there is one more portion of this graph which I didn't mention yet, and that is basal body temperature. All right, another Another factor that can be measured to essentially monitor where a woman is in these different cycles is body temperature. Note that in the first half of the cycle, in the follicular phase / proliferative phase, basal body temperature is a little bit lower than it is in the secretory phase / luteal phase. Okay. On the day of ovulation itself, there is a dip in the temperature as well. So body temperature gets lower and lower and lower, and then it's going to rise up for the remainder of the cycle. Okay, and note that these differences are not very big. We can see that it's like a fraction of a degree. And so, you know, there's a lot of maybe human error, and, you know, you have to be super precise when you're measuring these factors so that you can be sure that you're getting a good depiction of what's happening in her uterus and ovaries. Okay. I also wanted to point out some, what I think is kind of interesting data. Yet another portion of this graph. And this is, you know, same kind of thing. Here is the beginning, or day 0. Here's ovulation right in the middle, right? And here is the end of the cycle, so the beginning of menstruation. This line here is showing progesterone levels, and so that looks very similar to what we see in this graph here. Right, so this is from a peer-reviewed paper. It is averages of, you know, however many women were in this study. And so the other factor I want to point out here is the blue line, which is desire. And so just like we talked about with testosterone, estrogen and progesterone, as you might appreciate already, they can also really affect behavior, right? So not only emotional behavior, but also things like libido. And so there are receptors for these hormones in the brain, as well as on lots of different structures throughout the body. And so I want to point this out because it's kind of a cool connection, I think, evolutionarily. You should always be thinking about like why, or you can always be thinking about like, why did these things develop? Why did these different things evolve to be this way? And so here we know that progesterone increases pretty much right after the woman is most fertile, right? So the egg is ovulated here. It is only fertile for 1 or 2 days tops, right? So if she is going to get pregnant, if fertilization is going to happen, it's going to be right here. So by the time progesterone levels are super high, if she's going to be pregnant, or if she's going to get pregnant in this particular cycle, she already is pregnant. And so her desire is going to go down, right? She's not fertile anymore, right? Because here's progesterone. This is, in theory, supporting a developing embryo, if there can be a developing embryo. And so there's no immediate need to use resources on intercourse because the fertile period is already over. If we look back to that fertile period, we can see that desire increases over the first half of the cycle. Okay, and it kind of spikes right here in her most fertile time. Right, so again, this is just kind of a cool tie to evolution. Like, you know, biologically, the goal is to make more of yourself to pass on your genes and, you know, essentially populate the earth with your particular genes. And so even behavioral shifts, even though we might not think that humans are so controlled by what's happening in our body. We think that our mind can just like override absolutely everything, but there's a little more to it than that, right? We are biological organisms after all. Okay. And actually, keep that in mind, perhaps. We'll come back to this in just a moment. Okay. Any questions about any of that at this point? It's a lot to take in, I understand. All right, so this isn't high school sex ed, but I think that it makes a lot of sense to talk about different types of birth control strategies now that we know exactly what is going on within the ovaries and within the uterus. Okay, so of course, there are lots of different types of birth control strategies. I'm not going to talk about anything specifically, but I want to point out how some kind of bigger picture strategies work or don't work in terms of what's happening in the female. Okay, so the first one I want to talk about is the rhythm method, or the natural family planning method. So this is not taking anything, any kind of pharmaceuticals. This is not using a physical barrier like a condom. This is abstaining from intercourse on days when ovulation may be occurring, right? So all of these strategies are aimed at preventing pregnancy, right? So preventing sperm from meeting egg during the fertile period. Okay, so in theory, the days when the female is most fertile, that is the greatest chance of sperm meeting egg, is right after ovulation. Okay, and so in theory, this method can, of course, or using this method, a couple would abstain from intercourse during this ovulatory period. Okay, and so the way that this is done is that essentially the woman would monitor things like her basal body temperature, right? So it's lower, 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 dips down a little bit on the day of ovulation, and then increases after that. Monitoring things like cervical mucus texture. Remember that during ovulation, the mucus thins and promotes sperm entry as opposed to being thicker other times during the cycle to prevent sperm entry. And of course, you can do urine tests for luteinizing hormone. Remember, LH should only be high right before ovulation. Okay, and so as you might imagine, this is by far not the most successful strategy to prevent pregnancy. It has a very high failure rate due to the irregularity in women's cycles. So keep in mind that the second phase, right, the luteal phase, the secretory phase, is the consistent one, right? It's always going to be 14 days from ovulation. And so even if you know exactly the day of ovulation, right, so day 14, if intercourse occurred the day before, right, sperm can live for 1 to 2 days. Okay, so even if the woman ovulates here, the sperm can still be alive until over here. And so fertilization can still occur. And so this is the variable period. It can be 14 days, it can be 7, it can be 2 months, right? Very variable. And so usually once ovulation has occurred, right, the outcome is already determined. Okay. Textbooks will say that the ovulated egg is only viable for 12 to 24 hours. There are some extenuating circumstances, right? Maybe up to 48 hours, but really a very brief window. Of course, this is assuming that the woman is only ovulating a single egg from one ovary, not both. Also, it's assuming that ovulation is only occurring right once on day 14, not a few times throughout the cycle, right? So so many different factors, so many different variables go into pregnancy. And so that's why there is a very high failure rate. The other thing is that I guess another strategy for this rhythm method or natural family planning is that over 40% of men release sperm into their pre-ejaculatory fluid. So in the first male reproductive system lesson by Dr. Fink, you heard about the bulbourethral glands and how they release pre-ejaculatory fluid to essentially prepare the vaginal canal for the entry of sperm. They change the pH of the male urethra as well as the female vagina. It creates lubricant, etc. And so in a lot of men, sperm is actually released into this pre-ejaculatory fluid. Essentially, the muscle that is preventing sperm from leaving the ejaculatory duct is not completely closed, completely contracted. And so some sperm can get into the pre-ejaculatory fluid. And so pulling out is also has a pretty high failure rate. Right. And so some men don't do this and some men do, but without a microscope and some further investigation, it's a little difficult to know which one of those types of people you are. All right, oral contraceptives, of course, much more popular in society today. These pills have come a long way in the past few decades. Initially, there really wasn't much research done on these medications because it was kind of taboo to even be talking about them in the first place. And so not a lot of funding was thrown at this problem. And so initially, birth control pills were just like, you know, straight estrogen. We're going to give her as much estrogen as possible and kind of see what happens. And so there were some of these pills like DES, for example, that cause a lot of birth defects, and not just in the babies that were born, even when mom was on these pills, but also in their children. And now we're getting to the point where we might see in their grandchildren as well. And so, again, this has come a long way in the past few decades. And in fact, today it is the most effective form of birth control when it's used as prescribed, right? You can't miss a day or else that kind of throws off everything. Also, it takes a little while, of course, to change her uterus, change her ovaries enough to be fully effective. Okay. Essentially, these oral contraceptives are potent forms of synthetic estrogen and or progesterone, and the combination or individual hormones themselves is determined by your doctor, of course, but essentially, kind of the idea here is that these pills greatly increase the levels of estrogen and or progesterone, and they kind of mimic this portion of the cycle. Right, so this portion of the cycle is after ovulation would have already occurred, right? So it's actually kind of preventing ovulation because more or less her body thinks that she's already pregnant, right? If progesterone and estrogen levels are already this high, well, ovulation has already occurred. Maybe, you know, baby is already there, so we should support baby as a result. And so kind of the simplest way of explaining this is that kind of tricks your body into thinking you're pregnant, mimicking this portion of the cycle. Now, there are, of course, potential side effects to these pharmaceuticals. Not only is there an environmental impact, right? So these hormones are specifically designed to be super potent, right? Ethinyl estradiol EE2, for example, is a very common oral contraceptive, and your body physically cannot process all of that. And so a lot of it ends up being released into the urine. Your urine, of course, goes into the sewage treatment plant, and then those hormones aren't actually removed from the sewage, and they go out into the world, and they can actually produce a lot of feminization in the animals in the environment. That's actually what I studied in grad school. There are some wild impacts of oral contraceptives on the environment. But in the human herself, there are health risks. And for the most part, these are all tied back to, you know, what would happen if you were pregnant for 5 years, 10 years, you know, for the entire duration of taking birth control. So really, what is a high level of estrogen or progesterone stimulating on purpose, right? So what would baby need? Well, baby would need higher blood pressure to deliver more blood to the baby. It would affect how sugar is managed, et cetera, right? So all of these things are in some way directly or indirectly linked to more or less long-term pregnancy, right? So it can exacerbate existing problems like hypertension, any kind of fat or sugar metabolism. So, you know, gallbladder, diabetes, mellitus, like these types of things. Okay. Also, something I find cool And this is tying back to something we talked about quite a while ago now, and that is the MHC, the major histocompatibility complex. Remember in a lesson on the immune system, I talked about how each person has a unique combination of major histocompatibility complex genes. And so individuals with really similar MHC, right, are more likely related. And of course, you don't want to reproduce with someone who's closely related to you. And so humans and other animals have developed a pheromonal mechanism of communicating essentially how related you are to somebody. And so, essentially, you should be attracted to someone with a very different MHC so that your offspring might have a better chance of recognizing more stuff and being immune to more stuff, as opposed to, reproducing with your sibling and then your offspring, among many other issues, might not have the same strength of immune system. Okay, so remember that was a while ago. The pill. Remember that the pill is essentially tricking your body into thinking that you're pregnant. When you're pregnant, you, right, biologically, evolutionarily, are not supposed to want to go out and like meet people who are different than you. You're not going out to, find a new mate because you have just mated with somebody and you want to kind of pair bond with your family, your support system. So when estrogen and progesterone levels are high, right, and your body, thinks essentially that it is pregnant, a woman is more attracted to people who are more similar to her, have a similar MHC. Again, because there's a baby coming and, she needs to build up her support network with people who are closely related to her. And so this Scientific American article talks about how, or kind of nicely summarizes a study in which the researchers looked at the MHC between, couples with a woman on the pill and couples with a woman not on the pill. And it turns out that the pill makes women more attracted to people who are more closely related to them. Again, wild behavioral implications of different hormone levels. Of course, you always think of these hormones as having, maybe making women emotional at different points of the cycle, but there's so much more to it than that. So there's a link if you're interested. And, just a plug for Scientific American. It's been around and written by scientists for well over 100 years. So a great source of science news, Scientific American or Scientific American Mind here. Okay. They do not pay me to say this, but I think it's really cool. Okay. Last one I want to talk about, again, this is not an all-inclusive list, and this is not advice for anyone's personal decisions or anything. This is just like, here's how it works. Plan B, right? So hormonal postcoital contraceptives. This is essentially using hormones or other types of synthetic chemicals after unprotected intercourse, the morning after pill. And so this involves taking a type of contraceptive within the next couple days after unprotected sex. And so these work up to 5 days on average after the unprotected event. And so this is essentially before implantation could ever occur, right? So even if fertilization occurs, implantation is prevented here. Okay, so implantation is the actual embedding of the fertilized egg into the endometrium. Without the embedding, the fertilized egg cannot be sustained. Okay. In addition to preventing implantation, these pills can prevent the ovary from ovulating. Right. So even if the unprotected intercourse happened before ovulation, these can prevent ovulation from occurring, right? So remember that sperm can live for a couple days in the uterus. And so if they are already in the uterus, when ovulation occurs, then of course fertilization could occur. And so this prevents ovulation, and it also makes it really difficult for the sperm to fertilize the egg. And so for the most part, these contraceptives are preventing fertilization from happening to begin with, right? They're not eliminating viable eggs or anything. They're just preventing fertilization from occurring. Okay. Of course, the sooner these are taken, the better. So it reduces unexpected pregnancy rate up to 89% when taken within 72 hours. But the longer you wait, the effectiveness is going to decrease. Okay. So again, just a brief aside now that we know a little bit more about how these things work. All right, so I want to finish today by talking about fertilization itself, and then again, in our last class together of the semester, we will talk about what happens after fertilization. Okay. So fertilization itself is when the sperm's chromosomes combine with the chromosomes of the secondary oocyte. Okay. So not sperm meets egg, not even sperm releases genes into the egg, but genes from mom and genes from dad join together and then stimulate the mitosis event. Okay. As soon as sperm's chromosomes combine with egg's chromosomes, we call this a fertilized egg or a zygote. Okay. And so in order to accomplish fertilization, there are a lot of different things that need to happen. So every time I talk about this, I'm just like amazed at the fact that people exist at all. Like all of these things have to happen just right in order to make a baby. So our biological systems are really working to promote fertilization, but really a lot has to go right in order to achieve it. So first of all, sperm has to actually get to egg. After ejaculation, the sperm, right, those millions of sperm that are ejaculated, not all of them actually get to the egg, right? A lot of them leak out of the vagina immediately. They never even make it to the uterus. Some of the sperm within the vaginal environment are destroyed by the acidity. Remember, there's this kind of like fighting back and forth. Sperm really likes it alkaline. The uterus and vagina are, for the most part, pretty acidic. And so the acidic environment is going to kind of kill off the weaker sperm. Survival of the fittest here, right? Some of them don't make it through the cervix, right? Many of them do not make it through the cervix. And those that do make it through the cervix enter into the uterine cavity, which is, of course, acidic, and it is populated with phagocytes, right? So there's little Pac-Mans all over the place, like just waiting to get rid of sperm that have made it into the uterus. And so, of course, successful sperm will ultimately have to swim all the way up without eyes, that is, right? They're not seeing anything. They're just kind of like randomly going. So all the way up through the uterus into the fallopian tubes or the uterine tubes, and finally towards the ovaries. And so really very few sperm actually reach the uterine tubes to begin with. We're talking like 100 to a few thousand, even though millions were ejaculated. And so there's a lot of weeding out of different sperm. Okay. Also, the sperm themselves have to be kind of activated. Okay. Remember that there is absolutely no reason for sperm to be swimming inside the man, right? There's no reason for it. Sperm are propelled via peristalsis throughout the vas deferens, ejaculatory duct, and ultimately the urethra. And so sperm have to be capacitated. They have to be turned on and start swimming once they are ejaculated. Okay. So they're turned on, they start swimming. Okay. Sperm must be capacitated before they can penetrate the oocyte. So this is a kind of a pathway of events, but ultimately what this results in is enhanced motility, right? So not just like swimming slowly, but like really excited. The membranes of particularly the acrosome and the head have to become fragile. All right, so essentially what you want to happen is that the acrosome, the plasma membrane over the head has to like break down and become a little bit leaky so the enzymes can start to digest the zona pellucida of the egg. Okay, so there are chemicals within the semen that capacitate the sperm, but also the uterine environment is going to weaken the acrosomal membrane, right? So remember that it's pretty acidic, and so that's going to break down that membrane to make it leakier to help penetrate the egg. Okay. And the sperm, as I said, they follow this like olfactory trail. So they're literally like sniffing out the egg. We talked about this concept a little bit in the immune system, right? So different white blood cells can complete chemotaxis, right? So they can follow this trail of different cytokines to actually get to the scene of infection. Sperm are doing kind of the same thing. So they're like sniffing out, do we go to the right fallopian tube or the left fallopian tube to actually get to the oocyte? Okay, so let me show you this little animation here. So this is the process of fertilization. Then I'll break it down for you into steps.
When ejaculation occurs during intercourse, approximately 200 million sperm, or spermatozoa, are deposited into the vagina. They swim through the cervix, propelled by whip-like motions of their tails, or flagella, after which muscular contractions of the uterus direct them to the uterine tubes. This process usually takes between 30 minutes and 2 hours. Only around 200 spermatozoa will reach the secondary oocyte in the uterine tube, and of these, only one will fertilize it. Fertilization cannot occur until two processes have taken place: capacitation and the acrosomal reaction. These can take several hours. Capacitation is not fully understood, but secretions from the uterus wall and uterine tube destabilize the plasma membrane surrounding the head of the spermatozoa, or acrosome, resulting in the membrane becoming more fluid, which helps to prepare the spermatozoa for the events of fertilization. The spermatozoa become hyperactive. Their flagella beat more frequently and their heads move laterally. The capacitated spermatozoa move through the corona radiata, a dense layer of granulosa cells surrounding the oocyte, and come into contact with the zona pellucida. The zona pellucida expresses specific receptor proteins called ZP3, which bind to proteins expressed in the heads of the spermatozoa. The binding of ZP3 triggers the acrosome reaction, during which the enzymatic contents of the acrosome are released. These enzymes help to digest a path through the zona pellucida, allowing the spermatozoa to enter the perivitelline space and reach the plasma membrane of the secondary oocyte with which it fuses. To ensure that only one spermatozoon penetrates the zona pellucida and fuses with the oocyte membrane, fusion of the spermatozoon and oocyte membranes activates a fast and a slow block to polyspermy. During fast block to polyspermy, after fusion, the oocyte membrane depolarizes, preventing other spermatozoa from fusing with it. Slow block to polyspermy is also stimulated by this depolarization. During slow block to polyspermy, a wave of intracellular calcium is released, causing small cortical granules beneath the oocyte membrane to release their contents, rendering ZP3 inactive and making the zona pellucida impermeable. Upon the spermatozoon entering, the oocyte undergoes meiosis II and further develops into the female pronucleus. During this time, the sperm develops into the male pronucleus, and the two pronuclei fuse to form a single diploid nucleus, or zygote.
Okay. So let's break this down into steps here real fast. As the video just said, of course, the sperm are going to approach the egg. Here we can see the Zona Pellucida and we can see the corona radiata. The sperm are activated, so they very quickly beat their flagella to kind of wiggle their way through the corona radiata cells. Remember that the corona radiata used to be the granulosa cells of the follicle. Okay. So the sperm breaches the oocyte coverings. This is actually a pictomicrograph of a sperm wiggling its way through the corona radiata itself. So once this occurs, the sperm is going to bind to the Zona Pellucida, okay, which is going to trigger the acrosomal reaction. Right? So these membranes have already been weakened, and so it's really easy for them to dump out, or the acrosome to dump out the enzymes, which are essentially going to eat away at the Zona Pellucida. Okay, so the enzymes extracellularly digest holes in the Zona Pellucida, and remember that dozens, hundreds, maybe thousands of sperm are all kind of trying to digest the Zona Pellucida at the same time. It really is a race to the finish. As the sperm head digests the Zona Pellucida, it's getting closer and closer to the surface of the oocyte itself. Okay, the acrosome is then going to bind to the plasma membrane of the oocyte. And so we can see here that there are lots of little receptors that bind specifically to human sperm acrosomes. Okay, so the gametes are going to fuse, right? So the sperm itself and the egg itself, this is not yet fertilization. The DNA has not combined. Okay, in order to assure that one sperm fertilizes the egg, not 2, 3, 100 sperm, as the video was saying, there are a couple different ways that polyspermy is prevented. And so poly is many, sperm. So how does the entry, the binding of one single sperm, prevent the binding of all of the rest? And so what happens here is, again, as the video said, a couple different things. First of all, there's a cortical reaction. Okay, so these little vesicles here from the endoplasmic reticulum, right, they're already packaged up by the cell a while ago. And so in response to sperm binding to receptors on the surface of the oocyte, these vesicles are going to exocytose their contents. And essentially that is going to physically destroy the sperm receptors, right? So none of these other sperm are then able to bind to the membrane. Also, this is going to cause some osmosis, right? So essentially, this extra calcium and such is going to pull extra water into the Zona Pellucida and essentially kind of swell up this lining on the outside of the cell. And so any sperm that are still trying to get through the Zona Pellucida are going to have to go much farther in order to get there. So it literally like swells up the outside to further separate what's happening inside the cell from other sperm trying to enter. Okay. Once the winning sperm releases its DNA, right? It releases it as a little pronucleus here. And essentially this nucleus is going to be kind of, danced across the cell. As soon as that nucleus enters into the oocyte, meiosis 2 is finally going to be completed. So we can see here, this is anaphase. These DNA are going to be isolated into yet another polar body. Okay, just getting that extra copy out of the way. And the one single copy of DNA that we actually want for fertilization is going to be formed into the female pronucleus. Okay, and so the two pronuclei, right, the male DNA and the female DNA, are literally going to be danced across the cell in a process called amphimixis, kind of like approaching each other and, flirting around and then finally fusing together to create one single nucleus. Chromosome 1 from mom is going to join up with chromosome 1 from dad, chromosome 2 from both parents, right, are going to be matched ultimately. So now there are 2 copies of all of the chromosomes in the human genome, right? As long as everything worked out okay. Okay. As soon as the nuclei fuse and the chromatids start to organize themselves, we then call this a zygote. Okay. And so that is the end of this particular lesson.